Okay, this is what's got to be taken into consideration is the enzymes, which is the bacteria that's in your body. Bacteria are considered to be efficient enzyme factories. They can produce the right enzymes in the right amounts to digest food and, reduce odors and all that stuff. And they do it for your body specifically. Now, it's better said, bacteria are the factories that produce enzymes. They're the factory. So without the factory, you can't produce. So what does enzymes do? They're in the endocrine system, and they secrete, the endocrine system uses these digestive enzymes I was just talking about through a normal physiological process responsive to hormones involved in digestion. It's all about digestion. The process is separate from exocrine release. And anyway, the main key here is the endocrine secretion of digestive enzymes. Could have a bunch of different purposes in your body. It has the only thing that happens in your body chemically is always done by an enzyme, because chemistry cannot work fast enough inside of you unless you have the enzyme. I'm going to show you exactly how an enzyme works in a second. So they have physiological functions, enzymatic purposes, non-enzymatic purposes, immunological, self-monitoring. It does everything. It's all about digestion of your food, because if you don't have all these enzymes, you're done. And I just showed you bacteria are the factory that creates the enzymes. The enzymes come out in these little balls. We're going to get into that right now. All right, see this here, I just talked about the endocrine system. They work together. Your nervous system also works closely with your endocrine system. Now this is about the gut-brain axis. Everything is about the gut controlling something. In this case, it's the brain. In COVID, it's the lungs. But they work through the endocrine system. They all do, working with enzymes and hormones. So this is the gut-brain axis. Now we also have the gut-lung axis. This is where we get into the COVID-19, COPD, because this is a different exchange. This is the gut-lung is a bidirectional exchange of microorganisms. Microorganisms, the bacteria, metabolites, immunosignals between the gut and the lungs. Now, when you're inhaling, all kinds of little particles are coming into your lungs. Everybody's inhaling the same stuff. Why does this guy not get sick and this guy gets sick? Or why does this guy not get as sick as this guy? They call it like a pre-existing condition. Well, what does a pre-existing condition mean? It means, the guy's susceptible to getting sick. Well, why? My feeling is, and I have looked into this pretty closely, that COVID is associated very closely with pneumonia type problems and so forth. But I see all that as loose junction issues. And when I say loose junction issues, it's where your lungs are doing this all day long. They have to do this all day long. So the seal between your lungs have to be very, very good and stay. Otherwise you crack open like that, in comes the COVID. So I believe it has something to do with the bacteria to create the enzyme that creates the mucus and the seal. And I'll show you right now what I mean. All right, this is what I think they want to look a little closer, is the gut-lung axis. Now, this immunal signals and all that are sent between the gut and the lungs because of bacteria and enzymes. Now, it's facilitated by the gut microbiome, which is all the bacteria. It connects the two organs, the gut and the lungs, through the blood and lymphatic system. All of these little things, these ribosomes, go through the lymphatic system, and that's correct. And, of course, in the blood. Now, as well as the common mucosal immune system. That now is understood as the interstitial layer, the common mucosal immune system. That's kind of a new organ that they have discovered. Now, the gut-lung axis plays an important role in respiratory disease and can affect both the gut and the lungs. Well, the gut, normally I say the gut affects the lungs, and once the lungs get infected, then you got all kinds of issues, because I believe the bacteria that's affecting the lung has got something to do with the rubberness of the lung. The stretchability of the cells have to be able to move like this in the lung and have more of a junction seal. And you're continuously inhaling all of these pollutants right into your lungs. And that's where the transpiration of blood into the oxygen is, right into your bloodstream. So if you don't have a good tight seal in there, and you don't have a good immune system inside your lungs, you're invadable. That's what being immunocompromised is primarily about, is you can be easily invaded. And that's where it happens in most cases, is my take, is through those junctions in the lung that have to do this all day long. Okay, we all know that a lung has to inhale and exhale, and inhale and exhale, and inhale, and if you don't do that continuously, you die. So you're always taking in things, and you see these people working on all kind of dust and all kind of crazy things. They're inhaling all this stuff, and then you get in crowds of people, whatever they're breathing out, you're inhaling in. There's almost no way to stop it. Now, so what do you need inside your lungs to keep you protected? Well, you need tight junctions. All right, now, cell junctions in the lungs, called apical junctional complexes, are made up of tight junctions and adherens junctions. These, they can move, but they never get apart. They never separate. Let me show you what I'm talking about in an anatomical drawing. All right, I was going to try to really make this complicated, but there's no sense to do that. There's different types of junctions. Let's just talk about the types of junctions, where they exist in the body, and how they have to be able to move and so forth. Some of them don't have to move at all. Some of them have to expand and contract. All right, this goes back a ways, back to 2015. And they're saying there's some new players in the game about this functional layer. And I'm just going to cut right to the chase. I was just talking about tight junctions. If they're not tight, you're going to be in trouble. And the tight junctions are located more apically compared with the other junctions, and are multi-protein complexes. Multi-protein complexes consist of transmembrane proteins. Remember this, multi-protein complexes, transmembrane proteins are these tight junctions, and those are in the lungs. All right, here's the key to this whole thing. We know that there's junctions between the cells. If you breach through those junctions, you invade in the tissues. They're made as a membrane, sealed. Now, the intercellular junctions that form adhesive forces connect neighboring cells. They separate the external environment from the tissues. There are three main types of intercellular junctions. The tight junctions, which is basically in your lungs for sure, the adherens junctions, and the desmonas. Now, here's the key. The tight junctions are located more apically compared with the other junctions, and are multi-protein complexes that consist of transmembrane proteins. Here's the key word. Most people wouldn't understand this, apically. Well, what does apically mean? It means on the top bump, on the bump of the membrane. The tight junctions are located more on the bump of the membrane than the other junctions, which are down below. But these also contain multi-protein complexes. Well, what are multi-proteins? That means multi-enzymes. Enzymes are proteins. You see this? This is a question in science. Are enzymes proteins? Are proteins enzymes? Well, yes and no. They come from the same source, which is this trigger ribosome. They unsheath, and then they have two different duties. but they are basically, an enzyme can be a protein. You can look at it both the same way. I traditionally think of a protein as a building block and an enzyme as a destroyer, but they both are interchangeable. They just come from a different bacteria. One bacteria squirts out this type of ribosome, which makes an enzyme. Another one squirts out another one, and it makes a protein. It's exactly the same. So are they the same? Well, yes. You see, I show you the one with the trigger balls coming out of here. Those are ribosomes. Those are so tiny that you can fit literally millions of them in a single human cell. Millions of those tiny little balls. Now, where do they come from? They come from a single-celled creature, one cell, which is a bacteria. And you got trillions of these living in you. There's more bacteria in us than there is us in us. They run the show, and they really run the show. They create these ribosomes, and there is thousands and thousands and thousands of different species. Therefore, there is thousands and thousands and thousands of different chemistry sets. That's all that is, a chemistry set. When that shoots that ribosome out, if it's a free floater, goes through the whole body anywhere it wants, and it collects inside cells. And it's a chemistry set, and it's waiting to be activated. If it gets activated, it turns into the enzyme. So originally you have the ribosome. All right, the ribosome comes floating around. It's running around here and there, and all of a sudden something bumps into it, and it unsheaths. And that's the way it works. There's some kind of molecular trigger. Somehow it gets turned on. Let's just put it that way. When it does that, it turns into a gigantic molecule. The sheath falls off. That's the capsid, which is a sheath. And out comes all of this exotic stuff. And when I say exotic, I ain't lying. I ain't lying. There it is. That's basically what an enzyme looks like. Now, don't forget, it came out of here as a ribosome. So you got a little ribosome here, and I mean it's way smaller than this. Let's say the ribosome is like this big. Just tiny, tiny, tiny. And like I say, you can fit millions of them in a single cell, in what they call the cytoplasm, the inside fluids of the cell. They just floating around in there. Now, here's what happens though. When this guy gets called on to do his job, it just goes and it turns into a huge, huge chemistry set like this, and it attacks or does whatever it's supposed to do as that enzyme. Now that's the way these ribosomes being produced by bacteria, they can get up right through the blood-brain barrier just like nothing, just zip right up in there. You try to put that through the blood-brain barrier, it won't go. That's why you can't get invaded by, and they can't get a lot of good drugs. They want to get into the brain because they won't go up there. You know, there's a barrier in there that says, no, you can't come around here. It's too big. We don't allow that stuff into the brain. So this is the key. This is what an enzyme is. It comes from a bacteria, and it sits at the top of the membrane. I remember I talked about apically located. That means all of those enzymes are sitting right on the outside edge of this, apically located. So if something comes here to try to attack you, or there's food out here that wants to break down, it just happens. It's right at that surface. All right, I think I showed you this, but you can never see it too many times. That is the new fluid-filled highway, just discovered or posted about in 2018. And it's they call it a newfound organ, and it is. It's your immune system, literally, lives in that membrane. And it's fueled by bacteria, and the bacteria create the enzymes. The enzymes are the ribosomes. They can go anywhere in your body. They just free float around, and when they're needed, boop, there they are. You can have millions of those tiny little balls in one single cell in our bodies. So you have plenty of them to do the job if you have the bacteria squirting them out. That's what they do all the time. Blah, 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 blah. And bacteria can populate their own cells. They'll make just enough bacteria to do the job. They won't over proliferate normally. They'll just make enough to do what's needed to do the job. But once you kill the things that are protecting you, the new invaders come in. They have no limits. And then they take over. But normally you have enough guys that'll come down this fluid-filled highway and they'll attack them or kill them. But if you don't have the right bacteria and the right enzymes and the right chemistry, I don't care what you got in there, it's not going to happen. Then you get a cytokine storm. It says, all right, I'll throw bombs at them. I don't know, give them something, whatever you got down there, just throw it at them. And it gets all inflamed and hussed up and nasty, and cells are dying and all kinds of things. And yes, your body probably is attacking itself. But that would never happen if you had the right stuff. You just don't have the right stuff. You're throwing bombs at something that doesn't respond. Okay, my friends, the simplest atom that exists and the smallest atom is the hydrogen. And they say it has one big gigantic proton, and then, which is positive, and then one little tiny electron about 2,000 times smaller that is negative. And that's what hydrogen is. I totally disagree. I say hydrogen, instead of being this and one little tiny ball, this and positive and negative, it's the same charge, but one of them's gigantic and one of them's tiny. No, absolutely not. Throw that away. This is what it is. This has 18 and 25 or so dipoles. Each one of these is a magnet. Each one is a magnet. All right, and they glue together, and at a certain point, and it appears to be 1823, I'm thinking, is a proton. Now, you got to have a little additional charge for an electron. A proton is like a charged particle, and it wants to attract something. We want an electron to have an extra charge. I believe it could go both ways. I'm still working on this, but I'm going to tell you right now, it is 1823 or 1825. So I'm going right now with 1825 for the hydrogen, 1823 for a proton. But when you get a hydrogen, you're up a little higher, so you got a little extra energy there with that extra particle. And a proton is a little less. But it's not, certainly not this. Absolutely not. So this, no. This, yes. And 1823, and somebody said, well, how do you come up to 1823? Well, according to Millikan's oil drop experiment, he took oil drops and he measured them somehow. I can't remember how. This goes back 60 years ago. And he measured the weights. And he found it changed all kind of different weights, but it was the same change in multiples. In other words, it was 0.0005845 atomic mass units, and you could divide it. Maybe 10 of them changed this time, maybe 5 of them the next time. It was always a multiple of that number. So he surmised, which I agree with, he was breaking them up into different electron level amounts of electrons, basically. And so an electron weighs 0.0005845 atomic mass units, and you multiply that times 1823, you end up with 1 atomic mass unit. That's how I came up with it. Now, is it right? I don't know. And 1823, I believe that's how I got it. Well, I know I got it that way, but I can't remember if that's the exact numbers or not. But it's that. It's 1823 was my number. And it turned out to be an extremely interesting number in a place called Strong's. But anyway, that's the hydrogen atom is the tiniest. It's the tiniest. But for me, it's not just one. It's 1825 or so. And then you get up to helium. Instead of having 2 protons and 2 neutrons, you've got 4 times almost 2,000. You got 7,350, I think, something like that. 7,000 instead of 4. And as they get up here, you got bazillions and zillions and zillions. Because for every neutron, you're going to add 1824. For every proton, you're going to change it to 1823. So the periodic table has to change a little bit. And then we also have to take into account enzymatic functions and catalysts with the isotopes and the half-lives, because that's how the body works, is nucleosynthesis and nucleophilic substitution. And what I mean by that is catalysts can take these metals, anything they want. They can come in there and go brrr, and reconvert it just like that into anything they want. They can move it up the periodic table or move it down. It's the decay. The decayed order doesn't mean it always goes down. It can go up. I've shown that extremely clearly. All you got to do is look up isotopes, and you'll see there's going to be smaller isotopes, and then you're going to get to a stable, and then you're going to have bigger isotopes, and then you're going to see the daughter isotopes can move up the periodic table. They don't have to move down. Decay does not always go down. With isotopes, they can go brrr, chop it up, put it back together anywhere they want. That's what an enzyme does. Okay, I know it sounds kind of crazy. I start talking about bacteria and about life and all that, and I always end up with electrons. Every single thing there is made of these two particles. I don't care if it's your eyeball, your teeth, or a bug. There's nothing that exists that is not made of these two particles. Everything there is. So, speaking about life, speaking about Mars, speaking about the sun, speaking about the corona, is also speaking about those two particles. This is dipole electron flood theory. Until we can get to understand dipole electron flood theory, there's no understanding anything. Nothing is correct. There is nothing that can become correct starting from incorrectness. They don't understand what's below an atom. Now, above atoms is a different story. Below atoms, no. And this is where energy exists. This is where we have to understand energy, enzymes, bacteria. These are inside of the nucleophilic and a nucleosynthesis. That's where we get our most, that's where everything really happens, inside of atoms. We don't even know where 95% of the universe is. It's inside of every atom we look at. We never see it because it's inside. It's completely coated with things that will bounce back at us. So that's it for today. Thank you. I love you all.